So we are presenting the results of a phase 1 study evaluating the safety and the clinical efficacy of a T cell receptor or TCR-based cellular therapy targeting a human endogenous retrovirus E peptide in HLA-11 patients with metastatic clear cell renal cell carcinoma. So for those who are not familiar with the concept, human endogenous retroviruses are genetic remains of exogenous retroviral infections that infected our ancestors and incorporated into the human genome. So these elements are transcriptionally silent in normal tissue, but they can be re-expressed on the cancer genome. So HERV-E is one of these human endogenous retroviruses. And the basis of our study is work performed at the National Institute of Health that identified that HERV-E peptides are immunogenic in kidney cancer, and that HERV-E is specifically expressed in clear cell renal cell carcinoma tumors, but not other histological subtypes of kidney cancer or normal tissues. So this supported the notion of HERV-E peptides as an immunogenic therapeutic target for T cell-based therapy in this disease. So additional work from our group at the National Institute of Health established that when human T cells are transduced with a TCR reactive to HERV-E peptides, these engineered T cells acquire anti-tumor activity against HERV-E expressing clear cell renal cell carcinoma tumors in preclinical models. And this provided the foundation for a scale-up production of a GMP manufacturing, compliant manufacturing process to manufacture HERV-E TCR transduced T cells for clinical use, so they can be tested in clinical trials in patients. So we are presented here the first phase 1 trial of HERV-E TCR transduced T cells for the treatment of metastatic clear cell renal cell carcinoma. This was a dose escalation trial with 4 dose levels. The primary endpoint was safety, and HLA-11 patients with metastatic clear cell renal cell carcinoma, measurable disease that were previously treated with at least with anti-angiogenic therapy and immunotherapy were eligible for their participation in this clinical trial. So all these patients were treated at the National Institutes of Health, but the cells were manufactured at Loyola University in Chicago. So what happened is that we collected the lymphocytes for the patients, and then CD8 positive T cell were transduced with the HERV-E TCR with a retroviral vector to a high levels, and then these cells were expanded in vitro with allogenic feeder cells and interleukin-2 and 15 containing media. So we treated 3 patients in each of the lower dose levels, and then 6 patients in dose level 4. So patients in dose level 4 receive 5 times 10 to the 7 transduced cells per kilo of patient weight, so very, very large number of cells. So we show in this trial that manufacturing large numbers of clinical grade HERV-E TCR transduced T cells is feasible, and it was successful in the 15 patients that participated in this clinical trial, but 2 patients required a second apheresis to manufacture their product. So all the patients were treated with lymphodepleting chemotherapy with cyclophosphamide and fludarabine prior to receiving a single infusion of this transduced cell, followed by interleukin-2 administration for 7 days. So in terms of results, so the primary endpoint was safety, and we showed that the administration of HERV-E TCR transduced T cell is safe with a manageable toxicity profile, and we did not see any dose-limiting toxicity or off-target toxicities. Unfortunately, we did not observe clinically meaningful benefit for our patients, and only one patient achieved a partial response, but it was a very short-lived response. So most of the patients in this trial had disease progression as a best response. So we know from we had important observations from our correlative studies showing that HERV-E TCR T cells were detectable in the circulation of the patients, but at low levels, low to moderate levels, even in dose level 4, where we administered a very large number of cells. So these transduced cells pick up in the circulation 7 days after the infusion, but afterwards, low levels of detection were seen in some of the patients at later time points, suggesting that the persistence of these cells is very transient and dependent of interleukin-2 administration. So we know from other cellular therapy protocols that in vivo proliferation of the infused cells is very important for anti-tumor activity. And our group believe that the poor in vivo expansion of the HERV-E TCR transduced T cells accounts for the lack of significant clinical activity in this clinical trial. So we suspect that the manufacturing process that was used to generate these cells with a very prolonged expansion protocol and the use of interleukin-2 and 15 in the culturing media might have led to the expansion of terminally differentiated and exhausted cytotoxic T cells with limited capabilities of proliferation in vivo. So our group, in order to gain some additional insight on the composition of the infused T cell subsets, we are profiling transduced T cell from infusion products used in this protocol to treat our patients using single-cell RNA sequencing and insertion site analysis. So additional work, preclinical work in our lab is testing alternative manufacturing approach conducive to preferential expansion of T memory stem cells and naive cells that have the highest potential for in vivo expansion. And all this work will be very important to improve the efficacy of the next generation TCR and CAR T cell based therapies targeting immunogenic HERV-E peptides for the treatment of clear cell renal cell carcinoma.